First, I'd like to thank the organizers for inviting me to give the talk. I'm Qingyao from Zurich. I'm a bioinformatician and I work on PaxDb. And today I will tell you a bit what it does and the current development. So PaxDb is a database of protein abundance across organisms. And this data is recorded in a physiological or healthy state. And it is in the format of part per million. So it's absolute abundance as a ratio of the whole proteome. And the benefit of using the data is that we also provide a quality score that's based on protein interaction. So in addition to a technical quality control, we have also the control that is based on biological meaning. And PaxDb can be used as a reference for both common and rarely studied species. Currently, we have 170 species in the last update from last year, covering all the 3 kingdoms of life. And for each of the protein abundance, you can select a taxonomical level like mammalia and look at the ortholog abundance. With that, you can learn something about the stoichiometry and whether the protein has a conserved abundance range. One of the applications that we have used to answer a research question is with this revolutionary signal that we can find by aligning the protein sequences between fungi species and reference species. And what we found is that at the more abundance range of the proteome, the sulfur containing amino acids seem to decrease, which is found, this negative relation is found in one particular clade of the 170 fungi species that we studied, which indicate that in the past there might have been nutrient limitation in the environment that forced the species to remove the sulfur in the abundant side of the proteome. So the PaxDb data, they are derived entirely from public data sources, from either publications or the data repositories. And so far, we can include data that are already processed either in the form of protein quantity or peptide quantity, and with additional processing like protein ID mapping and normalization, we summarize the experiment into a primary data set with a score. And then we group the data sets based on whether they come from the same species or for higher organism, the same tissue. And then we integrate them based on a weighted average iteration. And with this integration, it greatly improved the proteome coverage and sometimes even would improve the quality score. So far, we have included only experiments with processed data. However, in the public domain, most experiments are only unprocessed when the authors upload. So this means that if we want to include more data, we need to tackle 2 challenges. The first is project selection to match the PaxDb scope. And the second is bulk data processing when it comes to data from different sources. So project selection, it's a tedious work if someone has to sit there and look at the description for every project. Fortunately, from the previous curation effort, we have already collected labeled data, which consists of title and abstracts, and then the positive label or a negative label of different reasons. And with this data, we can go through 2 different approaches. The first is with the OpenAI GPT query, and second is the topic modeling. With the GPT, mainly we tried 2 different models. And then we iterate the prompt based on each time what are wrongly predicted. And it's a simple answer, yes or no. And we particularly want to improve the precision because we want to make it more automated. So when the model says it's yes, nobody has to go and check if it's really yes. So at the end, we could, so at the current iteration, we could achieve around 66% of precision. And with the second independent approach is the topic modeling, when we were using open source language model to create embeddings on this title plus abstract to make it a numerical representation, and then reduce the dimensionality and do the clustering on these vectors. And after that, we already have clusters, which are representations of topics. And then using more classical text mining approach, keywords could be extracted for this context that can be make it more interpretable. So on the right, you can see of the label text, what are the topics that the model has extracted. And sometimes the keyword itself already would be related to the label, but generally the context is in this range. So with this, we could have around 67 to 73% of precision for the top 3 models. Which is much better than the simple keywords that is 23% or 27% of precision. And because these two approaches are independent, we could merge them. And if we just take the consensus of the output, we could reach 80% of precision. And that means from the currently available data in the Proteome Exchange, it would be about 600 new projects to process. And now we have the projects, it comes to the processing of the data. So there are several considerations when we are processing data that are not coming from ourselves. So first, we want to guarantee the data quality that is robust and reliable quantification. And then we also need to take care of the resource, how much time it takes to process memory, CPU, and also how much effort we need to put in to adjust parameters based on the metadata. I've tried MaxQuant and OpenMS. So with the OpenMS, if some of you don't know, it uses the top workflow, which you can build up your pipeline based on the modules that are already present. And then there are a lot of flexibility there for the file converter. However, it's only the thermal raw file parser that's present at the moment. And then for the search engine, you can actually choose any kind of search engine that has an adapter to the OpenMS. And then you need to set yourself the MZ tolerance based on the data. And with the MaxQuant, I have tried LFQ and whether include second peptide search. And then I need to adjust experiments in the MQ XML file. And then with that, I tried on 3 different DDA projects. The first 2, they are with the author's process data and the disclaimer. They are both processed with MaxQuant. And the second one is the dilution series. And so I will quickly go through the results. So the first project is with QExactive. It's with soybean. And I used the FASTA file from 3 different sources, which have a very different number of proteins. But I think it's because of the duplicated proteome of the plant. So at the end, with these 3 different FASTA processed with MaxQuant, the identified peptides are very similar. And I also tested the second peptide or LFQ in the middle. So the shape indicates whether there is second peptide, and LFQ is indicated by color. So basically, there's not much difference whether LFQ is used, which is expected. And then with the second peptide, we could use 20% more time to increase about 5% of the identified peptides. And so with the author, it always detects much more than my processing. I don't know if it could be contributed by also processing 9 more treatment samples, which help with the match between rounds. And the intensity correlate very well. Second experiment, 6 samples with LTQ. And here OpenMS was quantifying much fewer peptides. Oh, did I mention that? Yeah, with this one, the OpenMS couldn't process the file because the converter didn't work. And the second one, OpenMS was quantifying fewer peptides, but of all the quantifiable peptides, they were highly correlated in the intensity. And then the last experiment with the dilution series. The spiked in peptides, the 48 proteins, they are all detected by the MaxQuant, and it could confirm the dilution as well as the consistency of the background proteins. However, this again in the OpenMS, with the author's parameter of searching with mass tolerance and a grid of different mass tolerance search. Unfortunately, the target decoy ratio is always around 1, which means after FDR, there is no more peptide left. So it means somewhere else there is a parameter that probably I need to adjust, which I cannot find. So I come to the comparison. For my purpose of large batch processing, MaxQuant seemed to be more robust, and the preset parameters work very well. But I'm sure if someone able to work with adjusting parameter, OpenMS maybe also works, and it could give more flexibility of searching with different engines. And runtime depends on the engines, and it can be quite fast if you choose a fast engine, and it provides some intermediate statistics, while MaxQuant provides all the output in the output text file. So with that, I will like to thank all the members from Mering Group and Zurich, and colleagues from SIB, and for your attention.